Hello viewers, we are going to discuss the topic study of human karyotype normal and abnormal, which is of common interest in undergraduate as well as postgraduate zoology courses. Before going into the topic, I would like to say this initiative is taken up by a Zoological Society of Assam for the benefit of students studying zoology or life science. I would like to dedicate this presentation to my teachers, Professor Bipul Chandra Kotoki and Dr. Biva Goswami, who taught me zoology in my undergraduate study in Nowgong College with lots of love and passion. So, let us go into the topic. The learning outcome of this topic would be to describe the structure and classification of human chromosomes, what is karyotype and how it is obtained, different chromosomal bending patterns and its use, study of normal and abnormal human karyotypes. Now, let us see the basic morphology of chromosomes. Each human chromosome consists of two arms joined by the centromere. The two arms are termed as P for the shorter arm and Q for the longer arm. Centromeres are essential structures where the mitotic spindles attach during cell division. The centromere is a complex structure with mostly repetitive DNA that is associated with a trilaminar plate structure known as kinetochore. Microtubules attach to the kinetochore and help in directing the chromosome movements along the spindle. While the kinetochore structure and many centromere proteins that are essential for the spindle fiber attachment are conserved during evolution, the centromeric DNA sequences are not conserved among the eukaryotic organisms. In human, the centromeric DNA consists of large blocks of middle repetitive DNA, which is known as alpha satellite DNA, which is rich in adenine and thymine. The total length of centromeric DNA varies from chromosome to chromosome. The chromosome ends contain a special structure which is known as telomeres. Telomeres provide stability to the chromosomes by stabilizing the linear ends of DNA molecules and are essential structures for maintaining the integrity of chromosome. They contain simple DNA sequences TTAGGG repeated many times. It may be up to 10 kb at the end of each chromosome arm. Unlike centromere sequences, this simple telomere repeat is conserved throughout evolution. An RNA containing enzyme telomerase adds new repeat units to ends of chromosome to maintain the telomere length. Over time, decreased telomere function is thought to result in progressive shortening of telomeres, leading to senescence and cell death. Depending on the location of centromere, human chromosomes can be classified into three groups. When the centromere is located in the middle with both arms being more or less equal in length, the chromosome is called metacentric. In human karyotype, chromosome 1, 3, 16, 19 and 20 are metacentric or near metacentric. When the centromere is located off center with one arm longer than the other, then the chromosome is called submetacentric. Chromosome number 2, 4, 5, 6 to 12, X chromosome, 17 and 18 number chromosome is submetacentric. When the centromere is almost at the end of the chromosome with one arm markedly smaller than the other, the chromosome is called acrocentric. Chromosome number 13, 14, 15, 21 and 22 are acrocentric and are distinguished by presence of satellites or secondary constrictions. They are the unstained regions or gaps in short arm that contains nucleolar organizing genes. Such secondary constrictions commonly separate a small segment of chromosome called a satellite from the short arm. In such cases, the secondary constriction is referred as a satellite stop, since it connects the satellite to the chromosome short arm. And there is another type of chromosome that is called telocentric chromosome, in which the centromere is located at the end of the chromosome, but this type of telocentric chromosome is not present in human. Now let us see what is a karyotype. It is a micrograph in which the chromosome images are arranged in pairs in order of length and according to the position of centromere of all the chromosomes within a single cell. The chromosomes are aligned with the short arms on top and the long arms at the bottom. By convention, the chromosomes are numbered roughly according to their size, with the largest chromosomes having the smallest numbers. It provides several kinds of information, including the number of chromosomes present, number and type of sex chromosome, the presence or absence of individual chromosomes, the nature and extent of detectable structural anomalies. Now let us take a look at the history of chromosome nomenclature. From the beginning, it was recognized that standardization was needed in describing human chromosomes. Several prominent investigators met in Denver in 1960 reached a consensus in the formulation of a common system. Autosomes were serially numbered from 1 to 22 in order of descending length. These autosomes were classified into 7 distinct groups. The sex chromosomes continued to be referred as X and Y. In London Conference 1963, the previously identified 7 groups were designated by letters A to G. Secondary constrictions were also recognized. In Paris Conference in 1971, after discovery of Q bending by Kasperson et al. In 1970, with the ability to identify each individual chromosome's band on bending patterns, it became essential to incorporate latest developments into the existing nomenclature, resulting in Paris Conference. The major highlights of these documents are designation of bands in chromosome, introduction of mosaicism and chimerism, codes for different bending methods, and designation of heteromorphic variants. Paris Conference document published a diagrammatic representation of the bending patterns of each chromosome called an ideogram or idealized diagram. In order to identify individual bands, a distinct system of nomenclature was used. The chromosome arms were subdivided into regions that included distinct landmarks helpful in identification of each chromosome. The regions were numbered in sequential order, starting at the centromere and progressing towards the telomere for each arm. Here in the diagram, the white band designates negative bands, the black color designates positive band, and the striped appearance designates variable bands. Here the chromosome number 1 to 3 belongs to group A, 4 and 5 belongs to group B, Chromosome number 6 to 12 and X chromosome belongs to group C. Chromosome number 13 to 15 belongs to group D. Chromosome number 16 to 18 belongs to group E. Chromosome number 19 and 20 belongs to group F. Chromosome number 21, 22 and Y belongs to group G. Let us see how the stained metaphase chromosome appears under microscope at high magnification. In the first diagram, the bands in the chromosome are very much distinct. In the second diagram, the human chromosome karyotype is shown after proper arrangement. International System for Human Cytogenetic Nomenclature ISCN was formed to add subsequent information in human chromosome nomenclature as per the resolution taken in Stockholm Conference in 1977. All subsequent documents pertaining to nomenclature are referred to as ISCN, followed by the year in which the document is published. The ISCN 1995 was the result of several significant changes and technological developments in both in clinical and cancer cytogenetics. The ISCN 1995 document provides detailed description of human chromosomes, heteromorphisms and basic guidelines for reporting normal as well as abnormal chromosomes using various cytogenetic techniques. Now let us see different chromosome bending techniques. Paris Conference devised a 3-letter coding system. The first letter describes the type of bending. Second letter describes the technique or the agent used to obtain the bending pattern. The third letter to describe the stain used. For example, the G bending obtained by using trypsin and giemsa stain is described by the 3-letter code GTG. The G bending, which is reported by Summer et al. In 1971, here the metaphase spreads are treated with trypsin, which digests the chromosomal protein. Then it is stained by giemsa and observed under microscope. This technique preferentially stains the regions of DNA which are AT rich. So far, this is the most popular method used to stain a chromosome. Here in the image, a metaphase plate showing typical G bending pattern. The next technique is Q bending, which is described by Kasperson et al. In 1971. Here, the metaphase spreads treated with the dye quinacrine mustard. Fluorescent bending is observed with UV light microscope. In this technique, it was possible to identify all of the human chromosomes with certainty, and thus this discovery is one of the most important in the area of clinical cytogenetics. The image shows an example of Q bands. While Q bands was a remarkable discovery, the bending method had fewer drawbacks. The prolonged exposure to fluorescent UV light results in fading of the bands, making the technique unsuitable for extended microscopic analysis. And secondly, the exposure of fluorescent microscope is a costly affair in many laboratories. Now let us see the steps of chromosome preparation from blood. Here 5 ml venous blood is taken and added in a culture flask which contains phytohemagglutinin and a suitable culture medium. Phytohemagglutinin is added to stimulate T lymphocytes to divide. Now they are allowed to culture at 37 degrees Celsius for 3 days. After 3 days, colchicine is added to the culture media, which prevents formation of spindle apparatus and arrests the cell division during metaphase, and In the medium, along with colchicine, hypotonic saline is also added, which fixes the cells. Now, the fixed cells are spread into slide by dropping, and then the cells are digested with trypsin and stained with giemsa. Now the metaphase spread will be obtained and they are ready for karyotypic analysis. Another popular technique is R bending, which is also known as RHG bending. This bending technique results a reverse pattern of G or Q bends, means whatever regions are stained in G or Q bending pattern, the remaining regions are stained in this technique. Here, the metaphase spreads are heated at 87 degrees Celsius for partial DNA denaturation and followed by staining with giemsa. They are observed under light microscope, and this technique is mostly useful in identifying abnormalities involving the terminal regions of chromosomes, which are lighter staining by G and Q bending. They generally stains the regions which is rich in GC base pair. C bending technique or CBG, in which the metaphase spreads are treated with barium hydroxide and stained by giemsa. This technique basically stains the constitutive heterochromatin at the centromeres and secondary constrictions and rest of the chromosome with pale blue color, as you can see in the diagram. Another technique, silver staining, which stains the nucleolar organizer region on human acrocentric chromosomes. These regions contains genes for ribosomal RNA or proteins which were known early to stain with silver. Now, the rules of nomenclature. As per International System for Human Cytogenetic Nomenclature ISCN 1995, the main points to designate a particular band are the chromosome number, arm symbol, region number, and band number. To describe a karyotype, total number of chromosome including sex chromosome, comma, the sex chromosome constitution is written. To designate numerical abnormalities, say female Down syndrome or trisomy 21, is written as 47, XX plus 21, depicting an extra 21 number chromosome. Sex chromosome abnormalities are described first, followed by autosomal changes. Structural changes are designated by symbols. Say for example 46XY del 1 P tar 2 Q32 colon means a terminal deletion involving the long arm of chromosome 1. Colon indicates a break at band 1q32 and deletion of the region distal to it. The rest of the chromosome from 1p terminal to 1q32 is present. There are many abbreviations used to describe karyotype. Few are listed in the table. Say, to designate deletion, del is written. To designate inversion, INV is written. To designate translocation, small t is written. Now, let us see what are chromosome regions and bands in a properly stained chromosome. Each chromosome arm is divided into regions. This division is based on certain landmarks present on each chromosome. Landmark is a consistent and distinct morphologic area of a chromosome that aids in the identification of that chromosome. A region is an area that lies between two landmarks. Regions are divided into bands and the bands are divided into subbands. Number of each subband is placed behind a decimal point following the first band designation. Now let us see how to locate a gene in a chromosome. Here in the image in chromosome number 1, we can see there are two arms P and Q. P arm is having 3 regions and Q arm is having 4 regions. In P arm, region number 1, it has got 3 bands. In region number 2, it has got 2 bands, and in region number 3, it has got 6 bands. So to designate a particular band, we need to write first the chromosome number followed by the arm, region and the band. Here if we want to designate a particular band, say for example the red arrowed region, first we'll write the chromosome number, which is 1, followed by arm, which is P, then region 3 and the band 2. So it would be written as 1P32. Similarly, another region in Q arm, if we want to designate it, then we need to write as 1Q12. To designate a particular location in a chromosome, then we need to write as, here in the image, 1Q31242. Since it is located in the Q arm of chromosome number 1, then the region is 3 and the band is 1 to 4th region, band number 2. Now the band resolution. It is the total number of bands depends on how condensed the chromosomes at the mitotic stage. Since the cell division is arrested, many cells would be at different stages of mitosis. Say a 400 band resolution may correspond to chromosomes late in metaphase, or say in a high resolution ideogram, say approximately 1250 to up to 2000 bands can be produced in mid prophase. In the diagram, a same chromosome is shown with 3 different band resolution after staining with GIMSA. The first one has shown here with 400 band resolution, second one is with 550 band resolution, and in the third one is 850 band resolution. So if the resolution is more, the number of bands will be increasing. Now the types of chromosomal abnormalities. Chromosomal abnormality can be broadly divided into constitutional and acquired abnormalities. Again, the constitutional abnormality can be further subdivided into numerical abnormalities and structural abnormalities. In numerical abnormalities, loss or gain of whole chromosome occurs. Say in Turner syndrome, one chromosome is totally lost, whereas in Klinefelters syndrome or in Down syndrome, there is one extra chromosome. In case of structural abnormality, a part of chromosome may be deleted. In Karyotype syndrome, we see deletion. There is another type of structural abnormality which is known as inversion, in which a part of chromosome can be inverted. In translocation, part of chromosome may be located in some different location within the same chromosome or in some different chromosome, due to which chronic myelocytic leukemia occurs. In case of acquired abnormality, It occurs during life in a healthy individual confined in a specific tissue, say for example the tumors. Now let us look into the karyotype of a Turner syndrome individual. In this karyotype we can see that one sex chromosome is missing, so it can be designated as 45, XO. In this individual we can see short stature, short neck and lack of secondary sexual characters. In Klinefelters syndrome, if we look into the karyotype, we can see there is an extra X chromosome. So the karyotype may be described as 47, XXY. If we look into the phenotype of Klinefelters syndrome individual, we can see they are usually tall, slightly feminized physique, then they have a tendency to lose chest hairs, osteoporosis and some sort of breast development. In a Down syndrome individual, if we look into the karyotype, we can see that there is an extra chromosome in 21. So the karyotype can be written as 47, XY plus 21. If we look into the phenotype of a Down syndrome individual, we could see flattened face, broad and short hands, short necks and other associated characters. If we look into the karyotype of a Karyotype syndrome patient, it is seen that there is a deletion in short arm of chromosome number 5. If we want to designate this karyotype, we have to write as 46, XY, 5p minus. If we see the facial expression of the same individual at different stages of their life, right from 8 months, again at 2 years, 4 years and 9 years, we can trace the changing facial expression of the same individual. In this disorder, the child used to cry like a cat. Here, their larynx and nervous system is poorly developed. Usually, the children lose the cry by the age of 2 years. Their birth weight is usually low and they are having speech and motor disabilities. Inversion. It is a chromosome rearrangement in which a chromosome segment is reversed end to end. Here, inversion of chromosome number 9 is shown. To designate the karyotype, it has to be written as 46, XY, INV 9 P12, Q13. If we magnify the image, pericentric inversion is seen between P11 to Q13. The common symptoms include congenital anomalies, growth retardation, infertility, cancer and recurrent pregnancy loss. Chromosomal translocation is seen in chronic myelocytic leukemia patients. If we look into the karyotype, it is seen that from Q arm of chromosome number 22, a part is translocated in Q arm of chromosome number 9. So the karyotype can be written as 46, XX, T 9Q plus semicolon 22Q minus. If we look into the slide to see the cell characters, we can see that the number of granulocyte is drastically increasing. The early symptoms in chronic myelocytic leukemia are fatigue, left side pain, joint and hip pain and abdominal fullness. If we look into the statistics of chromosome abnormalities, around 2% sperms and 20% ova have chromosomal abnormalities. So, among 100 conceptions, there are 25% chromosome abnormalities. In every 100 pregnancies, there are 15 spontaneous miscarriages, 50% of which have chromosome abnormalities. Among 160 births, one baby is born with a chromosome abnormality. To detect chromosomal abnormality early in pregnancy, amniocentesis is performed. Here, the fetal cells are obtained through a needle inserted in the amniotic sac of a pregnant woman. From the cells obtained, karyotype is made and analyzed for any genetic disorder. Another test to detect early chromosome abnormality is chorionic villus sampling. Chorionic villi are wispy projections of placental tissue which shares the baby's genetic makeup. Sample of chorionic villi is removed from the placenta. The sample can be taken through the cervix or the abdominal wall. The test can be done as early as 10 weeks of pregnancy. Fluorescence in-situ hybridization or FISH. Here, the metaphase chromosomes are labeled with a fluorescent probe which is very specific. Here in the first image, FISH of interphase nuclei with a chromosome 21 centromeric probe showing 3 signals consistent with trisomy 21 is shown. In the second image, FISH of metaphase with a probe for telomere showing signals at the end of each chromatid is shown. So, a gene or any particular region can be detected by using a suitable fluorescent probe. So, we have come to an end of this presentation. If you would like to study further, I would recommend following books and papers. If you have any queries or any doubt, please feel free to write to me in the following email address. Thank you very much for your attention.